Today our topic is pancreas. As before we have discussed about the duodenum. So in duodenum we have discussed something about the pancreas, about the secretion of the duodenum that go in the pancreas and stimulate the pancreas. So I think we have to discuss it here. So pancreas basically it is attached with the duodenum and it is a heterocrine gland. What is meant by heterocrine gland? That it is having exo and endo both parts. That is known as a heterocrine gland. After that you can see here the structure of this is structure of the pancreas. This is a duodenum. As I discussed, it is attached with the pancreas. You can see it is attached with the pancreas duodenum. So this is a duodenum and this is a pancreas. If we talk about the structure of the pancreas, this is a tail of the pancreas. This is a tail of the pancreas and this is the body of the pancreas. This is the body and this is the head of the pancreas. Head of and this is the head of the pancreas. After that this one is known as the intercallected duct. Intercallected duct. This one intercallected duct. After that this one is known as a hepatopancreatic duct. Hepatopancreatic duct. And this one is known as common bile duct. Common bile duct. As we all know, bile is, you can see formation of bile, it's in a liver and the bile is stored in the pancreas. So this is a common bile duct from where the bile is coming. So that was the structure of the pancreas. Now if we talk about the structure of the intercallected duct, this one, if we magnify the structure of this one here, if we magnify this, basically this is known as the interlobular duct. Interlobular duct, not intercallected duct, sorry. It is known as the interlobular duct. Now if you magnify the structure of the interlobular duct, that is a major pancreatic duct. It is a major pancreatic duct. So here if you magnify the structure of the interlobular duct, it is something like this. Something like this. This is interlobular duct. Interlobular duct. After that, it is, as I discussed, it is also known as major pancreatic duct. And these both are known as intercallected duct. Intercallected duct. And these cells are known as echiner cells. These cells are known as echiner cells. You can see here this is intercallected duct. Major pancreatic duct is divided into interlobular duct. It is further divided into the branches that call the intercallected duct. Intercallected duct. Now in intercallected duct, cells are present called the enterocrine cells. In intercallected duct, this is a duct. Something cells like this are present called the centroechiner cells. These were the echiner cells and these are the centroechiner cells that are present in the intercallected duct. Now if we talk about the centrocrine cell, centroechiner cells, basically these are the principal cells that release the bicarbonate ions. This releases the bicarbonate ions. Basically intercallected duct is stimulated by secretin and acetylcholine. As before in duodenum I have discussed that the secretin will stimulate the pancreas to release the bicarbonate ion that will be released by the and centroechinal cells. So this intercollected duct will is stimulated by the secretin and acetylcholine. Right. After that, if we talk about the echinal cells, if we talk about the echinal cells, basically echinal cells are stimulated by the acetylcholine. They will release pancreatic juice that will go in the duodenum. They will release pancreatic juice in which enzymes are present. In pancreatic juice, enzymes are present. Pancreatic amylase. Before I've discussed local enzymes in the duodenum, that I discussed about the amylase, lipase, and all about maltase. So these are present here, that called the pancreatic amylase. If you remember before lecture, previous video, I have discussed all about this pancreatic lipase that will obviously we can say break the protein. After that, pancreatic lipase. After that, nucleus will break down the nucleic acid. Basically, echinal cells release proteolytic enzymes. It will release proteolytic enzymes. Now, what are proteolytic enzymes? That are having two parts. It is having two parts. Number one is the endopeptidases and another is the exopeptidases. Basically, proteolytic enzymes means that will break the protein, right? That will break the amino acid, that will break the protein. So it is having two parts. Number one is the endopeptidases. What is the endopeptidase? Now, endopeptidase basically it will break down the bond between the amino acid chain. Like this is suppose that this is amino acid chain. This is amino acid chain. Suppose that this is amino acid chain. Break the bond present between the amino acid. It will break that bond. Endopeptidases will break the mid bond present between amino acid chain. It will break the mid bonds. It will break the bonds present between the amino acid. That is the endopeptidase. If you talk about the exopeptidases, you can see by the name that endo means something that is inside, and exopeptidase which is outside. So exopeptidase will break down the bond of the terminal bond. It will break down the terminal bond. So that was all about the endopeptide and exopeptidases. Basically, the pancreatic juice will work at the 8 to 8.8 pH. It will work to the 8 to 8.8 pH. In echinal cells, mitochondria, Golgi bodies, and endoplasmic reticulum are present. So that was all about the pancreas.